Hello, today's problem is, below is DNA fragment isolated from the beginning of the gene, determine which strand is transcribed, indicate the polarity of the two DNA strands, and then give the sequence of bases in the resultant message RNA and its polarity. And if you know how to solve this problem, or if you don't know but you can always try to solve this problem, so you may stop video here, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. And here's my way how to solve this problem. So imagine that we have DNA, two strands of DNA, and I would show them in different colors, and here we would have message RNA in the process of transcription. And why I choose the same color for the message RNA as one of the strands, and this is very easy explanation. Here, one thing that you have to remember, that message RNA always starts with 5' end. So this is going to be, even when you look at such picture and you don't know where is the 5' end, where is the 3' end, and if you know that this is message RNA, you always know that this is going to be 5' end. And of course, this is going to be 3' end. And now we can also tell all the prime ends for the DNA. So this is going to be mRNA, and this is going to be DNA, two strands, and of course if we have here 5' end, and this is going to be complementary strand, so this is going to be 3' end here, 5' end here, 3' end here, and 5' end here. And as you see, our prime ends here and here and here and here would be the same. And that's why I use the same color for both these strands. More than that, if for example we would have on one strand, on message RNA strand, for example, A, 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 on this strand here, we would have the same bases, that would be adenine, 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 and on the other strand that here shown with red color, we would have complementary bases, and this is going to be T, T, and T. So as you see, these two strands are also would have the same coding, and this one would have different one. So this would be complementary strand, and we call this strand as template strand, and this strand would be coding strand. So this is going to be coding, and this is going to be template. And of course, when we have, for example, on the message RNA bases like uracil, on the coding strand here we would have thymine, because in the message RNA thymine would be substitute with uracil. And when we have different bases, for example, C, C, and C, of course here on the message RNA we also would have the same bases. And if we would have G, G, G here, we also would have G, G, G here. So all three bases would be the same, except thymine that would be changed to uracil on the message RNA. And on the template strand, we would have complementary bases that is going to be C would pair with, for example, G. So here we would have G, G, G. And here on the complementary strand, we would have C, C, C. Another important thing to know about message RNA that somewhere at the beginning of the message RNA you should expect to find three bases that is going to be A, U, G, and this would be start codon. And this is going to be orientation, so A, U, G would be 5' end to 3' end direction. Or we also should search for the G, U, A in reverse direction. So because we don't know in which direction DNA present here, and this is going to be two DNA strands. So we should look for these three bases, and in order to find these three bases here, of course we have to build a message RNA. And as you see, when we would find AUG here on the coding strand, we're also going to find A, T, and G. In message RNA, thymine would be substituted with uracil. And now we can start searching these two strands of the DNA for this sequence. And let's check the first strand that is on top. So we are looking for ATG, that is equal AUG on the message RNA. So ATG, we are looking for AT, so AC here, and ATG. There is no ATG in this direction, and also we don't know, maybe this DNA flipped over, so we also have to look for the GTA, so in reverse direction. So let's look for the GTA, and this is G, and so there is no ATG or GTA on the top strand. Let's search the below strand for the ATG, and here is A, T, and G. Let's go to the end, and there is no ATG, and GTA, GTA we cannot find, but what we did find, we find ATG. So what this tells us? And this tells us that this is going to be 5' end. So this is going to be 5' end here. So this is going to be 3' end. And of course, the second strand of DNA would go in the reverse direction. So this is going to be 5' end. And here is going to be 3' end. And also, we can name these strands. So as you see, this strand that has ATG in the 5' to 3' direction would be coding. So this is also going to be coding strand. And the other strand that is complementary strand would be template strand. So this is going to be a solution of this problem. And also you may have a question about why start codon is over this region, but not the first codon here at the 5' end of the message RNA, and this is because this region here is called 5' end untranslated region. So this fragment wouldn't be translated into the protein sequence, and here we can find Shine-Dalgarno sequence to which ribosomal RNA would bind, and translation would start only from the codon, that is start codon or AUG. So this is going to be beginning of the protein production, and not this very end of the 5' end. And this is all for today. Thank you for attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Goodbye.